This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 189, recorded on June 14th, 2012. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Today, we travel back to Glasgow, Scotland, where I have a conversation with junior virologists at the Centre for Virus Research at the University of Glasgow. You may recall on TWiV 188, which was just last week, I had a conversation with senior virologists there, and I thought while I was visiting, I'd talk to some of the postdoctoral fellows who work there as well. We have not had many conversations with postdocs here on TWiV, and since I was visiting at the invitation of postdocs and PhD students as the recipient of the Sir Michael Stoker Award, I thought it would be good to hear their thoughts about what they're doing and where they're heading in their careers. So stay tuned for my conversation with Swetha, Gavin, Vanessa, Mark, and Mary. I thought it would be nice, since I'm here, to talk with non-PIs, right? And just get your thoughts. So I'm going to just ask who you are and how you got here and what you do and, you know, what's troubling you. what holds for your future, that sort of thing. You know, just whatever you want to talk about. We don't have usually postdocs. Are there no PhD students? Are you PhD? No. You're all postdocs, right? We don't usually have postdocs, so I thought it would be good to represent the institute that way. And I thought I talked to all of you, and I thought you would be good for conversation. You seem to like to talk, all of you. So that's why I'm sure a lot of people would do well, but I couldn't have more than this number, so that's why I asked you. So thanks for doing that. So let's go around, and I'll ask you to initially just tell us who you are and your position here, and then we'll go back and explore. So you are?
So hi, I'm Swetha. So I'm a CDF.
CDF, okay. What's that?
So that's called a Career Development Fellow in the Medical Research Council terminology of hierarchy, which is equivalent to a junior postdoc, I think, in the university setting. So I just joined a year and a half ago, and I've just been looking at influenza research as my topic.
Okay. Next.
Hi, I'm Mary.
Okay. And you are a CDF?
I am a CDF. I'd never heard of the term before coming to the MRC. But yes, and I'm working on the cytomegalovirus.
Okay. Next.
Hi, I'm Gavin. I'm employed here as an investigator scientist, and I'm also working in the same lab as Mary on cytomegalovirus, and I've been here about 6 months.
Hi, I'm Vanessa. So I'm an investigator scientist, the same as Gavin. I think I've been here the longest out of everyone, and I'm working on hepatitis C virus at the moment.
And hi, I'm Mark.
Sit up straight.
Just getting comfy. I'm also a CDF, so I've been here a couple of years since I finished my PhD in New Zealand. And yeah, I've got another year or so left on my postdoc. And I'm working on hepatitis C.
And now I'd like to know, just tell us a little bit of your history. So Swetha, where you born and where did you get your PhD and all that?
So I was born in India, and I was raised in India. So I did my undergraduate in physics and my master's in physics with a specialization in solid state.
Solid state?
Yes.
Cool. That's neat.
And then I was wanting to look at what I want to do for the rest of my life. And so I really enjoyed astrophysics, actually. So that was what I had in mind. And then there was a lecturer in my college who was pursuing her PhD part-time, and she had just got into this new department in the university, the big university in the city, which had a biophysical department. So I think I went and asked her what she was doing for a PhD, and she said she was looking at the viscosity of a rat's tail. And it was absolutely amazing to hear that. And I think that kind of fueled my interest towards biology. I mean, I always liked biology in high school, but I never did, advanced biology in school. So it kind of fueled back that excitement of being able to apply physics in biology. So when I finished my master's, I had this choice of whether I wanted to do biophysics, which was an upcoming field in India, and or astrophysics. But then I didn't have high school biology, so I needed to get the training in biology and the knowledge, basically. So I went and worked in a biological institute, in a structural biology lab, which applies physics to biology. So it's still within my comfort zone. And it was absolutely exciting. There was a lot of knowledge to learn, but it was a good decision to switch the field and try to combine two disciplinaries to look at the same subject. And I think from then on, I've just stuck on to structural biology, learning different things all along the way. So my initial interest into structural biology was in TB, looking at structural proteins in tuberculosis, namely the chaperonins, which are involved. And then I came over to Glasgow to do my PhD. So I did my PhD in Glasgow at the Biomedical Life Sciences Division. And I did it more on the protein complex, which is the pyruvate dehydrogenase complex that's involved in glucose metabolism. But it was a biophysical and structural study of that. So it was really nice because it introduced me to a different kind of biochemistry and molecular biology and different techniques. And then this is the first time I've moved to virology that I've spent, because I've never done viruses before, and it's really exciting because it's a different level of looking at things, and I'm still employing physics and structural side of things. So, but at the same time learning a lot of the virus aspects and the virology side of things. So that's where things stand.
Okay. Mary, how about you?
Well, I've always loved science. It's always been something that I've been good at. So that's actually kept me in science, something that I'm good at. I tried to play golf recently and I'm not good at that. So.
Are you from England or Scotland?
I'm from England. Yeah, from the Midlands, just between Manchester and Birmingham, a little town called Biddulph. And there's actually a professor at the Beatson Institute here. He's from the same small town. I asked him for a job, but no success. And I did my PhD in molecular biology and biochemistry at the University of Wolverhampton under a new investigator there called Angel Amasilla. It was really good. I was the only person in his lab, and much the same as you, Vincent, I got all the hands-on sort of training and everything. I really enjoyed that. So after my PhD, I thought I would do a postdoc as the natural progression. So I came here to Glasgow and I worked on stem cells for 9 months. And it was part of funding from a company, so I had to present my work at a company conference. And then my contract ended abruptly, and I luckily got 3 months in another lab working on G protein coupled receptors. And then I saw this job come up for 3 years, career development fellow. Sounds nice. So yeah, I applied for this job, and I've been here for 2 years, just over 2 years.
So who's the PI in your lab?
It's Andrew Davison.
And again, your virus is?
Cytomegalovirus.
CMV, okay.
Yeah.
All right. Gavin, how about you?
Let's see, so I was born in Glasgow, and bizarrely enough, actually, my parents both worked here in the 70s, and I never thought I would end up working in this institute, so it's been quite funny.
They worked at this
They worked here, yeah.
Wow.
Yeah, a
Were
long
they
time ago.
virologists or?
Yeah, Yeah, but I wasn't, so I've never really studied any virology. So I went to Edinburgh in the early 90s to go and study molecular biology at university. And when I left, I actually got a job as a lab technician. And it was a guy setting up his lab. I was his first employee, and I was a technician for about 2 years. And then I was offered the chance to do a PhD in his lab, which was really good actually. And at first I wasn't sure if I wanted to do it, and somebody persuaded me when they said, "Oh, how would you feel if your boss got somebody in to take over your project, as a PhD?" And I thought, no way. So I did the PhD, which was really fun. So that was in intracellular RNA transport in fruit flies. And then I went on to do my first postdoc at the Human Genetics Unit in Edinburgh. And I worked in translational control of messenger RNA in frogs and xenopus. And I did that for 2 or 3 years, and then I got my own funding to have a position there for another few years. So I was a senior postdoc in that lab and stayed there for a while. And we were looking at different poly-A binding proteins and their effect in translation and early development. And from there I went on to go back to Edinburgh Uni and did another postdoc looking at muscular diseases associated with the nuclear envelope. And we did a lot of proteomics. It was quite an interesting project. And that got me into omics and a little bit to do with human diseases. And from there, I then, after 3 years working there, I came to Glasgow. And I changed tack a little. I got a job in a diagnostic lab in the health service, where we were actually diagnosing human diseases, and it was a referral lab, so it wasn't just a screening. We were looking at different, sequencing genes and using different techniques to look at human disease mutations. And then after 3 years there, and then I came here to take up this position as an investigator scientist.
So now you're independent.
No, I'm not. So investigator scientist is, I don't know how best it's kind of a sort of semi-senior postdoc position. So I work in the same lab as Mary, and my position is dependent on the senior investigators funding, but it's a sort of semi-open-ended position.
But you're on a day-to-day basis.
I work
on your own
quite independently.
You have you have experience, and you can figure out what to do.
Yeah, I see my boss sort of once every week or two for a wee chat.
All right, very good. Now, Vanessa.
Okay, so I was born in Yorkshire, near York, and then I came and did my undergraduate in Scotland because we were advised at my school that you should go to university at least 100 miles away. So I did that. You tick box, do as you're told. And my undergraduate was in biochemistry with microbiology, and I did that at St Andrews University. And I was lucky enough, I did my undergraduate project in Martin Ryan's lab, and he'd only just moved to St Andrews at that time. And he's a really enthusiastic guy, just great to work for. That was a great experience. And he had a PhD coming up, so between us, he sort of taught me into it while I was working in his lab. So I did my PhD with him on picornavirus proteases, which was so nice for me to hear your talk yesterday.
I know Martin well. Yeah.
Going back to my roots. And but that was very, it wasn't very pure virology. It was using viral proteases more as tools of more biotechnology aspects. So when I finished, I decided that I wanted to learn some pure virology and actually handle virus. And there was a position open here in Glasgow with Dave Evans, which was just a short contract. And I thought, that's great. That would give me experience to learn some virology and look for what I wanted to do next. So I did that. That was on polio replication. And then there was a position opened up as that was coming to an end with a new investigator in Dundee University. So that's also in Scotland. And it may sound like I intended to stay in Scotland, but it just happens there is a lot of virology here. And it wasn't intentional at that point until in Dundee. That was where I met my Scotsman, so now I'm sort of stuck here. But no, don't mean that. So as Martin put it at the time, I went over to the dark side and went to negative strand viruses. That was worked on respiratory syncytial virus replication. So that was very exciting because we were starting a new lab and it was a small group, and that was really interesting. And then my boss decided to return to the US, and I was looking for another job, and there was a position here within the previous incarnation at the MRC virology unit, as it was then, in the RSV group. And as an investigator scientist, it was a bit more independent then because they didn't have a program leader. So I applied for that and I got the position. And then since then we've been reincarnated, or there's been a lot of change, and we're now the Centre for Virus Research, and there's been a lot of changes, so I sort of took a sideways step at that point, and I changed to Hep C about 2 years ago.
So you have a similar position as Gavin in terms of sort of a quasi-independent.
Yeah,
right?
Yeah, you're more independent. You can develop your own ideas.
And your PI is?
Arvind Patel.
Who I don't think I met. Did I? Yes.
He was at
the dinner last night. Sure.
At dinner last
I did meet him. Yes, he was mentioned this morning as well. So for you two, you were rather experienced. I'll get to you in a second. I didn't forget you. I'm sorry. You're rather experienced. What is next for you? How long can you stay here? And if it's limited, what do you do next, Gavin?
I'm not sure if I know the answer to that question. I believe my position is semi-open-ended. So as I said, it depends on the investigator who runs my lab being here. So if he keeps passing his 5-year reviews, then I can stay. In terms of what would come next, it slightly depends on, I believe, what happens to this unit. It's about to become a university department and move out of the MRC. So our future depends partly on what happens within the university system and not the Medical Research Council. But the progression would be to become some sort of career track and try to start your own group, and whether that happens or not.
But that's what you would like. That's what you would prefer.
I'd love that. That would be fantastic. Yeah, I've been a postdoc for a long time, and most people my age have started running their own lab long ago.
So if an opportunity arose that suited
jump
you,
at that.
you would look at it.
Yeah,
right?
That's right. Yeah, but I'm quite new here, so I need to get some good results before that happens.
Okay, and what about you? Similar idea?
Yeah, similar. I mean, I had a taste of it. But as Gavin said, we're just not sure what the situation's going to be when we transfer to university, and whether the same, what has traditionally been the MRC career progression is still going to be available. We hope so.
Yeah, but if you were productive and something arose, a position somewhere else, you would look at it?
Oh yeah.
You would like to have your own laboratory?
Yes.
Okay.
No, I say that with hesitation because after I was a team leader, and that's a whole different ballgame, is learning. We do our science, so we're trained to do our science, and you're not trained to do the management side of things.
That's right.
So that comes as a shock, but I know that now. So
yeah, you have to. It's something you pick up. Nobody asked me that today at lunch, but you guys must know you pick up a lot of administrative and management skills on the job. There's no way to teach. You don't learn it like you learn PCR or tissue culture.
No, I've often thought that it's a really interesting point. You get, a lot of people are brilliant scientists, but that doesn't necessarily make them good managers, and it's not something that's part of your official training in a lot of places. So
maybe it should be.
I think it should be.
We do
we get
have
some.
That here.
Yeah, we get some.
We can go on management training.
It's not compulsory though, is
Please.
it?
It's not compulsory.
I'm sure you'd like to say a lot, but you don't have a mic, so here, let me hand this over to you, and you you can tell us about where are you? On where you came from. That's right. So are you from England or Scotland?
No, I'm New Zealand.
That's right.
So yeah, I'm from a small town in New Zealand called Whangarei.
That's where hobbits are from. Right?
Yeah, there's a few hobbits. You possibly wouldn't pick it looking at me, but
no, you're not.
yeah, no.
You're one of those trees.
Yeah. The bigger lumbering ones that stomp around. So yeah, well, I'm from a small town called Whangarei, and kind of got interested in science at high school. I had a really, really good biology teacher, and he convinced me to give biology a bit of a go. I guess I was always going to university. It's something I've always wanted to do, but I was a little bit open as to what I'd actually do once I got there. So I ended up going to the University of Otago, which is also in New Zealand, pretty much the exact opposite end of the country to where I grew up. But I needed a bit of a change and break out on my own. So I went down to the University of Otago, and I ended up doing a Bachelor of Biomedical Science. So this was a new degree that they just set up. I was actually, I think, the second year through. The second year that it had been established, and this was a degree that was aimed at being more multidisciplinary than your traditional BSc and microbiology or in genetics or in biochemistry. So I kind of had the ability to take subjects across all the kind of health disciplines, just depending on what I was kind of interested in. So that worked out really well for me. The other good thing about this particular bachelor's degree was that the honours year, which was associated with it, so the fourth year of the bachelor's degree, instead of being a more taught kind of traditional honours year, it was 100% research based. So I had pretty much a whole year in my undergraduate of working in a lab. And during my time at university, one of my lecturers, a professor called Frank Griffin, who was actually the head of the microbiology department at the university, I got on really well with him, and he kind of convinced me to do my honours project in his lab. So I was working on a mycobacterial disease of cattle and deer called Johne's disease. And yeah, it was the immunology department, so I was kind of looking at the molecular immunology of this disease in deer. And that year of that honours project went really well, so I got convinced to apply for funding for a PhD, and subsequently won that funding, and did a PhD in the same lab, which worked out really well. I got on really well with my supervisors. And I had a lovely project. I got to travel around the countryside, going to deer farms and taking samples, and did a bit of immunology as well, which I really enjoyed. So yeah, that all went really well, and got my PhD, and about, 3 months before I finished, I was like, oh God, I've got to think what I've got to do next. I had absolutely no idea. I knew I really enjoyed science, and I wanted to carry on, so a postdoc was a kind of obvious choice for me. But I also knew I wanted to get out of New Zealand. It was time for me to travel and see a bit of the world, so I was kind of looking at America and Europe, anywhere in there, anything to do with immunology. I guess my main interest that had developed over the course of my PhD and the honours year before that was looking at the pathology of chronic diseases, so and how the immune response plays a role in that. So that's kind of how I decided that I'd actually apply for my current position, which is a CDF, but I'm working on hepatitis C. So it's quite a big change of field, really, moving from veterinary immunology to virology. But hepatitis C is a chronic disease as well, so I decided that it would there'd be related themes that I'd be quite interested in, and I applied for a job advert that was posted on the internet, sent off my CV, and ended up having an interview over Skype. And so this was at like 11 o'clock at night in New Zealand, which was, I don't know, maybe 8 or 9 o'clock in the morning over here. And I had the interview with my, my position is shared between the two hepatitis C labs, so I'm co-supervised by John McLaughlin and Arvind Patel. So I had an interview with both of them over Skype, and that finished about midnight in New Zealand, and I was just packing up because I actually had to come into work to get a good internet connection, and I was packing up, getting ready to go home, and I got a phone call straight away, and they'd offered me the position. So a month later, I was on a plane over here, and 2 days later, I was to start work.
How long have you been here now?
2 years now. So everything's gone pretty well, really.
Can you give us a sense of what you're doing in the lab?
My project's quite clinically based. It was an area that both John and Arvind wanted to expand into. They had quite strong basic science programs, and in Glasgow there's a lot of clinicians who treat a lot of hepatitis C infected patients, and they get on really well with the clinicians, but they never really had a kind of direct link between the clinicians and the patients who are out there and the basic research that's going on. So I was one of a group of 3 or 4 people that were brought in, new positions that were created to kind of bridge that link and bring some of the clinical work that's going on in Glasgow and link that up to basic research. So at the moment, I've got a couple of projects going on. One of them is looking at alternative splicing in liver biopsies from patients. So this is doing RNA-seq, deep sequencing of RNA species in liver biopsies. And I'm trying to identify changes in alternative splicing that has possibly been driven by hepatitis C, by the actual virus infection itself. And then also looking a little bit at the different metabolic dysfunctions between patients who are infected with genotype 1 hepatitis C and genotype 3 hepatitis C. So this is based on the observation that the genotype 3 patients get a higher level of steatosis, or steatosis is associated with genotype 3 hepatitis C.
So you're trying to find out if there's any particular gene expression?
Yeah, and how the different viruses are actually changing the lipid metabolism in the patients. So I've kind of set up a couple of patient cohorts, and I spend a lot of my time collecting blood samples and processing blood samples, and hopefully in the next 6 months or so I'll start doing some of the lab-based work and get some results from it. But certainly just a simple collection of samples takes a long time. It's a big process. So I spent a lot of time on that so far. And
that's why a lot of us don't do that.
Yeah.
It's important, but it's very hard to do.
Yeah.
So when you're done, what do you do next?
I don't know. I do enjoy science, and I enjoy what I'm doing. I like to carry on my research into chronic infectious diseases. I wouldn't mind staying in hepatitis C if the opportunity arose either. And I certainly would like at some point to have a go at setting up my own lab and having my own group. I guess it's just a matter of finding the opportunity. It might be a bit too much of a jump at the moment, so I think the next step for me would probably be trying to get a fellowship, so secure my own funding for a postdoc position, and then following on from that, try and move into setting up my own lab.
So do you think, I don't know if this is correct, but do you think that the British or Is that the right word for where we are? British? Yeah. Won't offend anyone. Is British just PhD system, not PhD postdoc system, not have you ready for a position after the first postdoc? Or is it usual to do more than one? And because it sounds like the general trend is that, right? You say you're not ready to do your own lab at this point.
I think I probably I think it would be a big jump, but I think I'd be able to do it at the moment. But I don't think my publication record would get me the opportunity yet. Okay. So I think one of the things that I think looks really good in terms of your career development is the ability to secure your own funding, independent of a PI. So I think that's quite a valuable step if you have some evidence or if you're able to secure your own fellowship position. It certainly is it helps in moving on to the next level. Ideally it would be straight into a permanent position and setting up my own lab. I think the more realistic option would possibly be doing a fellowship first.
Where do you think that would be? Would that be here in Britain, or would you go back to New Zealand or somewhere else?
Yeah, I guess that's a $1 million question. I mean, the great thing about science is you can travel. It's very easy to move to a new place. You can pretty much the whole world's your oyster. It's the one profession that you can go to pretty much any country and they'll open all the doors to let you in and work there. And the problem comes when you actually want to stay somewhere in particular. So when you want to settle down somewhere, then it actually gets quite difficult because obviously the pool of jobs isn't as big as some other industries.
The key is not to want to settle down.
Yeah. Certainly New Zealand, it would be nice to go back there someday. I think there's quite a strong culture that people go overseas to get the experience, to gain contacts, and just to be exposed to a bigger scientific community than what is present in New Zealand. But New Zealand does have a very strong scientific community itself, and certainly at some point in time I'd like to go back there and kind of give something back.
That's a good idea.
Yeah.
So let's hear from Swetha. How is that pronouncing your name right? Swetha?
Yeah, Swetha.
That's correct. Just tell us about what you do.
So I'm looking at influenza. So
Parrots?
No, MDCKs. We're still with the MDCKs. So I work with David Bella here, who has a structural approach towards understanding structure of viruses.
I just spoke with him, yeah.
Yeah. So Dave moved into influenza just a few years ago, so it's kind of his baby as well. I mean, it's quite normal for him to do it. So we both kind of sailing in pretty much in that same boat. Like sometimes it helps, like you were saying about you going to Peter Palazzi's lab, where he knows a lot of influenza stuff, and you say he's your PI and he knows it, so you follow it. But in my case, it's slightly different. We both are like in the same boat and we don't know most of the things. So it's more talking to other people and learning the stuff. But what's really nice is that because the thing is that I work in the CVR, and even if you work in one virus, everybody knows so many things. I mean, you're effectively in a virology department, and all the senior colleagues know so much of virology, and it's like a community here. Nobody stops you from going and asking somebody what, if they knew and you want to clear a doubt. It's really nice.
And people are willing
very
to
friendly.
help.
Yes, absolutely. So I think that's very unique about MRC centres. They function as a family unit, so it's quite nice to get the benefit out of that. So I think I've learned a lot from the time that I've been here, learning a lot of viruses that way, talking to people, colleagues and other PIs, and just learning the techniques and just clarifying things. So in terms of the research, what we'd like to look is obviously look at structure, but influenza is a very pleomorphic virus, so you cannot look at the structure that easily because you get a lot of different morphologies in a prep, and it's difficult to most of the structural biology techniques out there require a homogeneous population of a particular sample. So what we employ cryo-electron microscopy as a major tool looking at structure, and with the advance over the past 20-30 years, cryo-EM has introduced the tomography aspect of using to determine structure to a certain resolution. It's not really high resolution, but you can still look at it in a real single molecule sense because you're looking at one particle or one virion and then looking at it.
In 3 dimensions.
Yes,
right?
in 3 dimensions. So it pushes in that perspective. But personally, I feel the real power of tomography over single particle cryo-electron microscopy is the fact that you can see it in the context of a cell, which no other structural technique till date can do it. I mean, apart from confocal microscopy and the optical microscopy, which is different, but the resolutions are nowhere close to electron microscopy. So we all start off with the ultimate goal of looking at structure, but then you have to correlate it to function in the context of a cell. There's no point looking at it on its own and then saying it can do this or may be involved in this. So it's really nice that you can have a technique to see it in the context of a cellular approach. And tomography is a great technique for doing that. So what I've been trying to do is to look at influenza on its own, just a purified prep, and trying to see if I can get a slightly higher resolution structure by tomography, but also looking at its entry and exit mechanisms in our cell culture systems for the moment.
By tomography as well?
By tomography as well. And using other techniques like immunofluorescence, confocal microscopy, and so on to complement data. But the tomography offers you a great tool to do that because you can see virus coming in, you can get a snapshot of it just on the cell surface, see what are the virus cell factors that interact and so on. So you can pull out a lot of information with that kind of data. And it's very exciting times. There's so much of technology development on that end that another 2 decades or something, it's going to be absolutely amazing that you should be able to get fabulous data from it. And it's very exciting for me because I've never done EM before, although I've done a lot of other structural techniques. And it's a great feeling to be, there are times when you do your PhD and you're purifying a protein and things like that, and you see it on a gel and you think, yeah, that my protein's there on the gel, but it's a great feeling to see it visually and you know that, oh wow, that's how it looks. I mean, it's an amazing feeling.
That's why when we did this cryo-EM, we were very excited.
Yeah, it's a completely different feeling, I think. So I think, it's really nice. But what we'd like to do is to also understand these mechanisms in actual clinical samples. So we're trying to tie up with the hospital here in Glasgow to see if we can get some clinical samples and then image them on the microscope to see if we can look at the same kind of mechanisms. But we work on influenza, but not all aspects of influenza. We're particularly interested in the filamentous aspect of influenza, which all clinical strains seem to exhibit, and therefore it's thought to be clinically important, but all lab-adapted strains don't show these long filaments. So the virus, when the cell gets infected, seems to produce a lot of these filaments, perhaps taking half of the energy to produce these filaments, and nobody really knows for sure what they really do in the cell and how they're involved in the infection cycle or in pathogenesis and so on. So it's a very exciting time for us to be in the influenza field when people are starting to ask these questions, and there's a lot of technology out there that might help you understand these things. I mean, not obviously EM doesn't answer everything, but if you had a combination of techniques, then you might get to that final thing of what it really does inside.
So is one of your goals to do a single particle structure of a filamentous form? Is that possible?
Yeah, I think the way you can do it, you can get around doing a single particle, is by doing what's called subtomogram averaging, because you can never get a prep that will be homogeneously filaments, so that you can do a filament and then actually go about the reconstruction of doing a single filament. So the way for pleomorphic viruses, you do tomograms, you do several tomograms, and then you take the area of interest that you want and you assign them as a single particle, and you take enough of them and then go about the classical approach to build a 3D reconstruction. It's a lot of effort, but if you do it systematically, I think you can push the resolution to get a filamentous structure to about 10 angstroms or 8 angstroms.
Because it would be interesting to see what's inside them, right? Because
yes.
I saw one of his images of Semliki forest virus, and you can see there were 4 genomes in it, for example. So it would be nice to know what's in these filamentous particles. Maybe there's a cellular component that helps it to
absolutely.
be extended. So that would be really exciting to do that. It's very neat. What's next for you after this? You probably want to have a lab of your own, right?
I do. I've always wanted to do it, but I realized that over time, I mean, considering the current climate, it's getting tougher for younger scientists to step that pedestal and know. And I think if you want to be in UK and Europe, like the discussion we were having, it seems to be a little bit more difficult, because there's a large contribution of publications that seem to play an important role on that. And I think because I've moved I think I quite enjoy being virology now that I've been in 1.5 years. I think I really enjoy. There was a time when I thought bacteriology will be the thing I'll do, but I think since the 1.5 years and getting to know the system and things like that, I think I really enjoy it. So I think I would like to be in virology, and if I did have a lab and go on, I think it would be virology. I'm still not sure which virus. I mean, influenza still fascinates me, but I would like to know what else is there, just, because if you're in structural biology and you confine all yourself to one virus, it can be a bit risky, so it's kind of good to have a bit of a backup thing there. But based, just like what Mark was talking about publications, I think when you're trying to get a permanent position at the moment, you need to have a fantastic publication record, even when it's fellowship as well. So I guess I would be tempted to carry on, like trying to get as many good publications for another 1 or 2 years perhaps, and then maybe if I feel I'm ready mentally at that point and I've sorted my area where I want to hold my own lab, then I think I'll try for a fellowship, because I think it's quite hard that at the moment that most universities do not have so many permanent positions advertised, and the eligibility criteria, if you see, for these things are enormous. I mean, it's absolutely amazing how much they expect out of you for just coming into a lectureship or something these days. But
does anyone come out of a postdoc, the first postdoc, and get that sort of job here?
Quite rare.
They used to, I think.
Yeah,
it's very
We
rare.
have had one recent start that has done that.
Yeah.
But it's rare, very rare.
They used to be less rare, you think?
Yeah, I think so. I
Yeah.
mean, talking to the older scientists about their careers and what happened to them, then yeah, I think so.
Like my PhD supervisor, she didn't even do a postdoc. She went directly from her PhD to a reader, to a lecturer in the university, and she was great. She fitted the area that time. They were looking, sometimes you got to be in the right place and the right time as well. And it's the same with David Bella. He came as an electron microscopist. He's never done a postdoc in his life, so it sometimes has worked in the past, but I think over the present it's becoming that's very, very hard.
Well, I'm glad to see that despite the difficulty, you're persistent, all of you, so that's good. And you should be, if you really like to do it, you should be. Mary, what's your project on CMV?
So I've been developing a quantitative real-time PCR assay that can detect multiple strains of human cytomegalovirus from DNA extracted from clinical samples. And we hope to use it to monitor the complexity of infections and also to look at strain dynamics over time in these patients. So with a previous, I think he was senior investigator, got some ethics together. So we've got a cohort of 40 patients, liver transplant, kidney transplant patients. Thanks for the help, Gavin.
You can say I don't know.
Kidney transplant patients, and we're collecting bloods and urines from those, and we're taking multiple samples from those over a period of, I think, 2 years. So we've got pre-transplant samples and post-transplant samples. So we're going to use those samples to look at the strain dynamics. So
why do you use transplant patients for this?
Well, they have the theory is that they're more susceptible to harbouring multiple CMV infections. So we really want to look at whether there's a interplay between sort of the major and minor strains of CMV, and whether the minor strains disappear completely or whether they come back and they take over. And so it'll be an interesting.
And this is because they're immunosuppressed for the transplant,
exactly.
right?
Yeah.
So do you have control? Do you do controls of non-transplant people?
Well, we haven't actually, but we have the donor and the recipient, so we have them.
The organ donor.
Yep. So,
of course,
in
if
theory,
it's a liver, you don't have the donor anymore,
it's
right?
kidney.
It's kidney. Yeah, because it's a liver. Forget it. They're not going to live.
Yeah, that should have clarified things. So so in theory, they won't be well, they shouldn't have a high. If they do, then the poor recipient is going to be.
So the donor could have CMV.
Yeah.
The recipient could also have CMV before the transplant.
Yeah.
So these two, and you would see this happening if the donor gave an infection to the recipient by your assay, right?
Yeah.
Plus, you would see if the recipient developed additional strains, right, as a consequence of the immunosuppression. And what's the time course? Do you look over long periods of time at the transplants?
I think it's 2 years we have, maybe. But I think it's every 4 months that we're getting samples. So, but I didn't set up the ethics of the project, so I don't know exactly. Just focused on my assay for the past.
So right now you're trying to perfect the assay, right?
Well, it's perfected.
It's done
now. Yeah.
So now you're looking at samples and.
Yeah, we're just starting to process the samples actually.
Okay.
So we've just got a technician in the lab, because it started off that our lab had about 6-7 members when I first started, and then everyone became made redundant and retired, and it was just me. So it's quite nice to have a technician and also Gavin in the lab.
You have to have someone, otherwise.
Exactly.
There's no one to complain to or learn from or teach or whatever. Yeah. How much time do you have left?
Just under a year.
And then? What's next?
There's the possibility to extend our contracts by 11 months. So there's that option. But I don't know. It was quite a disruptive time when all the people left the lab.
Sure.
It was quite difficult, so I'm not sure exactly what I want to do on my next phase.
Do you feel you're ready to be an independent investigator?
Some days.
Very good.
Some days
more
not.
days yes and more days yes than no?
Depends. I think it goes almost week to week, really.
Yeah.
Because, I mean, like 90% of your experiments tend to fail. So those weeks are hard. But
if someone offered you an independent job, in 6 months or a year, would you take it?
if I got offered it,
Yeah.
yeah.
But
That's the easy option, though. Someone
Sure.
offers it to me.
But if not, you would continue to work and, as everyone else has said, accumulate papers.
I think so, yeah. I don't feel confident enough to have my own lab, particularly since I've changed subjects so often.
Yeah.
I mean, I've had 4 different subjects. I mean, that is good in a sense, because I've gained a lot of experience, different techniques, different backgrounds, but I'm not sure if that's really helpful to get in your own lab, because I just from looking at people that do have their own lab, they pretty much stayed in the same research area.
Right.
And they've sailed through that way. So I don't know how easy it is to have this sort of convoluted changing of different subjects to get to have your own lab. I don't know, it's a bit daunting.
Mm-hmm. But you want to do it, right? You're not considering alternative careers? On some days you are, right?
Yeah.
Because that's something I haven't heard. In the U.S., if I did this with postdocs, a lot of them would be considering alternative careers, like
Really?
business law, writing.
Wow,
I haven't heard that here, but it's I don't know, you would consider it if you felt that you weren't ready at any point, right?
Yeah. Well, to be honest, I don't know if there's that many resources that tell us what we can do. Like, there's industry, and then...
Well, you could switch entirely. You could be a lawyer. You could get an
no.
MBA and go into business. You could do science
if I was
writing.
rich, maybe.
You could do science writing. So there are a number of other things
Yeah.
you can do. You could work for a journal and then piss off scientists.
We do. We have a career development forum here where we invite people from different science careers. So we did have a science writer, but there aren't that many science writing jobs out there either. So it's a really difficult climate at the moment to look at other jobs. I mean, you'd almost be silly to want to change career paths at the moment, just because they've got so much competition of people that actually have experience. So.
Okay.
Yeah.
That's it. It's good. It's a good sense of the obstacles you face and what's available out there. Gavin, how about you? What are you doing?
So my project's kind of very complementary to what Mary's doing. So we're hoping to be able to actually sequence the virus, the CMV, from clinical samples.
The same samples she was discussing?
Yeah, some of the same samples. So I've been setting up to do that. It's been quite an interesting time. So this new technology that we've got in deep sequencing is going to enable us, for the first time, hopefully, to be able to look at that. So most of the times when people have actually sequenced the genome of viruses, it's been from virus that's been grown and in culture in cells, and very quickly when you do that, you find that all these strain dynamics disappear, and the virus very quickly adapts to growing in cells. So we've been setting up our new Illumina sequencer and getting everything, and I've actually gone back to learn command line Unix and all these things that I haven't used for like 15, 20 years to do all the deep sequencing analysis and get that set up. So we're hoping to be able to take the tiny amounts of DNA that you get in clinical samples and pull the virus out and then sequence it from really small amounts, and then build the genome, the entire CMV genome, which is it's big. It's, I think, the biggest genome of any virus. It's 230, 240 kb. So build the genome de novo from our fairly short reads, and then look at the different strains that we can see in there. And then the ultimate goal of the project is to look at different strains and see if they have different outcomes clinically for the patients. So I'm sure you're aware that many people carry CMV. It's a very common virus, and normally it's fine. But when you're immune suppressed, or if you for HIV patients, and also if you contract it in utero, it can be quite serious. So, and there's very, very little known about that at the moment. So that's the project that we're hoping to do. It's fairly early days at the moment, and I'm sort of still in the setup phase. We're just starting to get our first samples through the machine now.
So Mary, you'd mentioned that there are multiple strains of virus, and so you would pick that up on the sequence, right?
Yeah, we would be able to see the different strains as sequence variants. One thing the deep sequencing isn't so good for, of course, is to look at linkage. So it'll be difficult to say, you have this many strains. You'll just see lots of variants at different positions in the genome, and you might have to do that another way if you want to get into kind of saying, which variants are linked.
Because the sequence runs are short, right?
That's right.
And it's hard to associate. If you find a mutation in a particular fragment, you don't know which other mutation it goes with.
Exactly.
And there are bioinformatic ways to get around that, but it's difficult, right?
It's difficult to do that, but of course there's other techniques out there to look at that in different ways. So
you get overlapping fragments too. You can use that to try and, right?
The reads aren't really long enough, when you're looking at a big virus and genes that can be quite far away from each other. Your reads at the moment, they're getting longer and longer, and they used to be 36, then 50, and then 76, and now we can do 150, and soon it will be 250. But you're not getting back up to Sanger sequencing lengths of 800-900 base pairs.
Yeah.
So it depends. Yeah.
So I think I asked you what was next for you already, right? We talked about that?
That's right,
'Cause
yeah.
you said that you're not ready.
Well, you
to
know,
have
I'm the classic case, I think, of a totally unplanned career.
It's okay.
That's just, it's
I
just
like
gone
that.
along like that. And I've done lots of different things, and I've worked in lots of different systems. And I'm not a virologist. I was brought in here because I had some experience in doing sequencing and patient samples and looking at sequence variants. Obviously, I've worked with RNA, and I've worked with fruit flies and frogs and human diseases and all kinds of different things. I do love working in a lab. I think it's a really fascinating job to have. You've got these incredible tools, and you can discover these really interesting things. It's a real privilege to be able to do. So I want to keep working in a lab.
You're not considering alternative careers?
I'm not at this point, no.
And someday,
not.
I think you said earlier, you'd love to have your own lab someday, right?
Yeah, absolutely.
Okay.
Yeah, I would. I really would. Yeah.
Okay. Vanessa, what are you doing in the lab?
Over the last 18 months or so, I've been involved in setting up the high throughput screening system.
Not high throughput sequencing. This is
No,
something different.
Screening. This is something different. Yeah. So we started. It started from a reporter system, a reporter cell line for hepatitis C virus that they developed in the lab. So this is a system when the virus infects the cell. There's a construct in there that contains a reporter protein which is secreted into the media, and you can measure how much virus replication is occurring within the cell by simply removing the media and measuring the level of this protein. So we were interested. So when I first started in the lab, this sort of just got this cell line, and we were interested in using it to see if we could develop, because it was so simple and straightforward an assay, rather than doing lots of TCID50s or plaque assays that you would do traditionally for a virus. Whether we could transform this into a 96 well plate assay, so a simple assay. So that was the idea, and we developed that, and to start to develop that, we took a library of kinase inhibitors, because we like to maximize what we're doing, and one of the focuses in the lab is virus entry, and we were interested in the signal. We're starting to get interested in the signal transduction pathways that are going on within the cell. So we thought if we use a kinase library and knock out some of these pathways with inhibitors, give us some idea of what's going on. So we started with that in the HCV-CC system, which is done at containment level 3. In the UK, I think that's different from the US. And
it's not the highest level, right?
No, it's not the highest
4
level.
Is the highest, right?
No, yes. So it's in between. It's in between standard laboratories. And at this stage we were doing this all by hand, so hand pipetting. And I quickly realised that if you don't want to develop repetitive strain injury, you need some other machine that's going to do this for you. So we applied for some funding, and we were lucky to get a simple pipetting machine that could do 96 wells in one go. So we got quite excited by that, and it was like, oh well, we can do more now. So we'll test a bigger library, and we got a library of just over 1,000 compounds, and these are known drugs, to see if there's any that would have any drugs that are already out there that have been approved for use.
So these are all approved drugs?
70% of the library are approved drugs, and to see if these would have any effect on HCV. And also, as we're interested in the entry system, HCV entry is quite a complicated, well, any virus entry, but is quite a complicated process. There's lots of steps involving binding of the virus and then interaction with receptors, and HCV has a lot of receptors and co-receptors, then the uptake and the fusion steps, and there's a lot we don't understand. So we also thought if we find new compounds that disturb this this process at different steps. This might help us understand the mechanisms and what's going on with the virus that lets it get into the cells. So that's about where we're at the moment. We have another system which we use just to look at the entry step, which is a retrovirus-based pseudoparticle system which expresses the glycoproteins on the cell surface. So that part just mimics the entry process, so we can just focus in on that. And that has the benefit for us as well here that can be done at containment level too.
And you put that through the high throughput as well with the 1,000 compound screen?
We did the 1,000 compounds with that first because we couldn't take the machines down to containment level 3, so we sort of switched it around. Ultimately, we have just secured funding for a bigger machine. We're getting bigger every time, and eventually when we move to our new building, the idea is to have that equipment down in containment level 3, so we would be able to use the full virus.
Sure, because they're doing the pseudovirus screen first, biases.
It biases, it focuses on entry.
Sure.
Yeah, and ideally we would like to look at the whole thing, but it's practical. It's practical aspects. You know, we don't have hoods big enough for that.
And then the idea is you find compounds that inhibit specific steps and use those to probe those parts of the infectious cycle.
Yeah,
right?
That's the idea. And with the added benefit that maybe you could get potential. There's always potential for antivirals if you could detect
Absolutely,
that.
yeah. And these would be targeting. Well, they could, in theory, target cell processes that inhibit the virus as well, right?
Oh yeah. I mean that's sort of where we're at the moment. We divide. We can divide things up between what we think is hitting the cell, some cell factor, and we use other viruses as well that use the same entry pathway to try and dissect how specific these are.
I think we'd also already talked about your future plans, right?
Yeah.
Before?
Yes.
Now I forget what I've already talked about so far. So we've covered everyone. We talked about your future plans as well. So I think that was a nice discussion. Is there anything else anyone wants to talk about? We have a lot of people listening. This is your chance to talk about anything you'd like, anything on your mind.
Yeah, okay. So CMV has little awareness. I'd just like to emphasize the fact that the simple process of washing your hands can stop the transmission of CMV, especially important for pregnant women. And if they're around toddlers or children, wash your hands and it will reduce your risk of getting a new CMV infection. You know, congenital CMV can cause hearing loss and other neurological disorders. So yeah, basically.
What should you wash your hands with?
Soap and water.
Soap and water is fine. You don't need to use an alcohol-based
no.
thing. Or
No,
virus is quite sensitive to salt.
Yeah,
okay.
Yeah.
It's a good general idea anyway, but especially in those cases.
Yeah, there's a CMV, Stop CMV in the US, which is a foundation to raise awareness. And also we've just got a charity here called CMV Action, which has just got a charity status. So that's raising awareness as well. But it's amazing how many people don't know about CMV. So.
Good, that's great, thank you. Anything else?
I'm afraid I can't say the same thing for flu.
Yeah, of course. For all our viruses, you should have good hand hygiene all the time. All right. Well, I like the discussion. I appreciate it. I think it's clear that it's hard to do science here. Maybe at some point I should have a similar discussion in the U.S. with postdocs and see how that would compare to it. That would be really interesting. But yes, you want to? Yeah, here, absolutely.
Yeah, I guess there's been a bit of. I mean, we've talked quite a bit about the future of what we want to do and things like that, and I think these kind of things, these kind of forums, like these podcasts and things that you're doing, really good for kind of raising awareness of science in general. And I mean, science is quite a difficult career. I think that's the one thing that everyone will agree on. and it's only getting tougher at the moment. But I think we all do it just because we enjoy it so much, and you take a lot out of it personally, even if you don't quite get the recognition that you'd really desire from your career. It's certainly not as high profile, or you certainly won't make as much money as doing other things. But I think if you've got a passion for science, it's well worth sticking at it for a while and giving research a go. It's something you find out pretty quickly whether you are keen to stay in it, and also whether you're any good at it. It doesn't take a big commitment to figure out, if it's something that you want to do for a while longer. So, don't get put off by by possible bad prospects. It's there is a lot of good things about science. It gives you a lot of freedom and a lot of independence to answer questions that you're interested in, and I think that's something quite unique with science.
Yeah, we're enthusiastic about science on this podcast, but we never go behind the scenes and tell people how hard it is to become a scientist. So that's one reason to talk with you, and I'm going to do one in the U.S. with some American postdocs. Actually, they might not be American. They will probably be from various places, but they may have a distinctly different experience. But I think this was useful for everyone to hear that aside from PIs, it's hard, but we have dedicated people who are interested in doing science, as you say, and that's what keeps you going. So people should appreciate that, because science makes everyone's lives better. So they have to know that behind it are people that work hard and often find obstacles. So that's important. So I want to thank all of you for joining me today. Vanessa, thank you so much. You're welcome.
Thank you.
Gavin, thank you for joining us.
Oh, you're very welcome. It was fun.
Mary, thank you.
Thanks.
Hope you have more good days.
Yeah, me too.
Swetha, thank you very much.
Thank you for this invitation.
Yeah.
And Mark,
Yeah, thank you very much. It's been a pleasure.
it's been great visiting, and thanks again for doing this. I appreciate it. Thanks so much for listening to this episode. I have to apologize for the sound difficulties. We had a pretty noisy room, and some of the levels were quite low, and there was a lot of noise. And I'm not the greatest sound engineer, as you know, especially on the road. So I hope it wasn't too distracting for you. As you can see, being a postdoc is a tough time. Traditionally, it's the time between when you finish your PhD and when you have an independent position. It's a period when you learn how to run a lab on your own. However, you mainly do experiments. You don't have any training in administration of any sort. And as you could see from the individuals we spoke with, some of them don't feel they're ready, even after a single postdoc, to run their own laboratory. I think it would be really interesting to interview some postdocs from the U.S., where traditionally you do a postdoc and then you get a job. Although in recent years it's been harder and harder to get jobs out of the first postdoc, and many individuals are doing more than one. But my overall impression, just having worked in the U.S. for many years in science, is that one postdoc is often sufficient to get a job. And it may be because, in part, in the U.S., the years of a PhD are not limited. As you heard in the UK, you have only so many years to finish a PhD, and that may reverberate through the postdoc years in terms of you being ready to be an independent investigator. I'll be in Madison, Wisconsin for the American Society for Virology meeting in July. I believe it begins on the 20th or 21st. If any postdocs in virology will be there, seek me out. Send me an email ahead of time. I'd love to sit down with 3 or 4 of you and record an episode about you and your thoughts. And we can see how they compare to the experience of postdocs in the United Kingdom. And by the way, you may have noticed that I didn't have enough mics for everyone in Glasgow. I had to hand mine over to Mark back and forth, and so that accounts for some of the noise. Once again, I apologize for that, but I hope you got the idea from the discussion anyway. As always, you can find TWiV at iTunes, the Zune Marketplace, at twiv.tv, and at microbeworld.org/TWiV. Over there on Microbe World, we also have an app for your iPhone or Android device that you can use to stream the episodes to your devices. As always, send us your questions and comments to TWiV at twiv.tv. You may be wondering if we plan to discuss the Fouché manuscript on H5N1 influenza virus transmission in ferrets, which was just released this past Thursday. I wasn't able to record a TWiV because I was traveling. I was in Seattle Thursday and Friday. But we plan to discuss it on our next episode, TWiV 190, which will be recorded this week. So stay tuned for that discussion, and that'll be recorded as usual in the TWiV studio, and hopefully the sound will be better for you. I want to thank all the postdocs at the Center for Virus Research at the University of Glasgow who participated in TWiV, and I particularly wanted to thank all the students and postdocs there for nominating me for the Sir Michael Stoker Award, which brought me there in the first place. So many, many thanks. I'm honored, and thanks for doing a TWiV with me. I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.